For the introduction. So yeah, I come from the same lab, CREATIS, which focuses on medical image analysis in Lyon. So this Okay, super. Okay, so this morning I think you've got the basics of machine learning, mostly supervised, and I hope that you're now able to answer this question, like learning, what does it mean? Learning, basically, it means you have a model. For example, a line that classifies between the blue and the orange samples. And learning means you optimize the parameters of your model, okay? So if your model is a straight line, there's two parameters, like the slope and the intercept, A and B, so you have two parameters to optimize. So you may start with a bad guess, like a horizontal line, and you look for the best coefficients that best classify your data. So you try to change A and B to optimize their value for solving the problem. Okay? When you have a neural network, that's the same thing. So this is the different neurons that are connected. So you have a weight that links the combination of the input values to the next neuron, etc., to try to solve this problem, to try to classify again the orange and the blue. And here the decision line is more complex, and you have much more parameters to optimize, which are the weights between the neurons. Okay, so that's the learning for a more complex case. And obviously, learning, it depends on your data. I hope you're all aware of this. If I have few data, maybe this would be my guess for the classification line, a straight line. And actually, if I have more noise in my data and more data, I may try to have a better guess, which is a more complex line. Okay? So this could be a wrap-up from this morning's session, the basics of learning, the basics of supervised learning, and try to keep in mind what is learning about. So this afternoon, we'll look for what is called self-supervised learning. Sometimes people call it unsupervised learning. So the basic thing, if you remember from this morning, when you go for supervised learning, you have labels. Labels means you know which data are blue and orange, which data are healthy or diseased. When you go for unsupervised learning, why? But maybe I just picked all the data in the hospital, all the images of the brain, for example, from the hospital, without any label, without knowing the age, without knowing the disease, and I just want to look for things in the data. So I don't want to use the labels, or I don't have any label. In that case, I go for unsupervised learning. The other thing is, maybe my labels, they are not relevant for my problem. So think of heart failure. Heart failure is not just binary. There's not, maybe you could say some people are healthy and some people are diseased, but actually it's much more complex. Across the course of the disease, you may fall into a simple or a benign heart failure, if we can say this, or a more advanced problem. And actually there's kind of a continuum, kind of a spectrum of the disease. And in that case, you may say, it's not relevant to use labels. I may go for a continuous stratification of the risk into the patients, for example. So that's two kind of questions to keep in mind if you want to go for unsupervised or self-supervised learning. And in the title of the talk, quick disclaimer, I won't talk too much about that, but there's semi-supervised learning. It means you may use some data for which you have the labels and many data for which you don't have labels. So think of this problem. If you only use the labeled data, you may say, okay, that's my classification line. But you could say, okay, apart from these two patients for which I know this one is healthy, this one is diseased, I may take all the images in the hospital and actually find a better structure into my data, and then my classification line would be that one. Okay? So many questions to answer. The thing of interest, and I think it could go towards your own applications, even if you're not into medical imaging, there are these ones. Basically, you may want to identify subgroups or patients or materials that have similar behaviors. You may look for anomalies with respect to a given set of data. And then other considerations regarding the way you apprehend the data. So I'll start with these ones, very famous applications of unsupervised learning, which are clustering and anomaly detection. Clustering basically is this. You have a cloud of points without any labels, and you would like to find automatically, so think these are all unlabeled. You would like to say, this is a group of patients that are consistent. This is a group of patients that are also consistent. So there's many algorithms for this. That's one, which is k-means. You start by saying, I may have two groups, so I choose a number of clusters. And then across iterations, I optimize something which is the position of the center of my clusters. So you see across iterations, I'm trying to move these big blue dots and this big red dot until automatically finding consistent data. So back to the learning problem you've seen probably this morning. Here, the parameters are the things that you optimize are the centroid of each cluster. And then you may say, I have an energy to minimize, a loss function to minimize. And in that case, you want the clusters to be as consistent as possible. So you would like to minimize the within cluster variance. That's one example. What for clustering? One example, you would like to do segmentation, even if this is kind of outdated. But you would like to find automatically in this image consistent patterns of similar intensities. In the last presentation, just before, we had also, or maybe the previous one, we had an image of, I think it was the titanium texture. And you may say in the titanium texture, I would like to identify 3 or 4 kind of grain levels that are consistent. So you may go for unsupervised segmentation with clustering. So in that case, you work with the histogram of the intensities, and you may get this. The other thing we do for medical imaging is, based on some data, could we find a space where each patient corresponds to a dot? And then we would like to find groups of patients that are similar to each other. So, for example, based on cardiac imaging data, on that application, we may identify 4 groups. And then if we look at each group, maybe the green group is low risk for a given therapy. Maybe the red group is high risk for a given therapy. And then this might be useful for the clinician. So in medical imaging, popular algorithms for clustering are k-means, Gaussian mixture models, many examples. The question that we may keep in mind is that, okay, it's nice to find clusters, but what if a patient is at the boundary between clusters? Is this patient here? Should we think of it as a blue or a green patient or something in between? So may we question these boundaries and think of a gray zone? And the key point is we cannot go for clustering directly on the images because they are very complex. They are what we say high dimensional. There's potentially one information at each pixel. So there's many information. And we should find a way to reduce the dimensionality of these data. Basically, the step is encoding, and I'll come back to it afterwards. Okay, so we should first process this data, reduce the dimensionality to find a space where we can apply the clustering and this kind of things. The other application, and I think in your field of interest, you may have the same issue, is can we automatically detect anomalies? And if we think of anomalies, something which is abnormal, you have a reference. You have in mind what is a normal material, what is a normal behavior to something, what is a normal patient. And then you have a new material, you have a new patient, and you would like to say how much it looks compared to the reference population. That's anomaly detection. So the first thing, it corresponds to my diagram before. You start with images or data. You encode them. Again, we'll come back to what encoding means. You reduce the dimensionality such that you have a representation of your data. Here, each dot is a data. Each dot is a patient. Each dot is a material. And if you have a new data, then you could say, in that space, can I look how far are my new samples from my reference population, and then get a score. If it's far, then I'll say there's a huge anomaly probably. That's the first thing to work. So if some of you did some mathematics or are into the field, a way to compute distances between the samples, there's a simple one, which is called the Mahalanobis distance. It's very intuitive. In one dimension, it's just the distance to the average divided by the standard deviation. So if you take a red dot here, I say, what's the average of the green? What's the standard deviation of the green? And basically, I'm saying the red is that much distance from the average, and I weight these by the standard deviation. Okay? In multiple dimensions, in 2, 3, 4 dimensions, that's the same formula, just instead of standard deviation, you have the covariance matrix, and then you have a vectorial formulation for the distance to the average, but that's the same thing. Another way to see anomaly detection, still unsupervised, is this. You say, I try to encode and decode my data, namely automatically knowing a representation of my normal data. So think of, imagine I'm able to do this. So you could think of encoding and decoding as, for example, JPEG compression, image compression. So you have a complex image, you encode it to a few bytes, namely that's your compressed JPEG or PNG file, and then you are able to decode to reconstruct a high quality image. So if you do this with healthy images, then you learn how to reconstruct healthy images. So suppose now I have a deceased patient. If I encode this patient and I reconstruct this patient, I won't be able to reconstruct a deceased patient. I only learned what a healthy patient is. So I will only reconstruct a healthy version of my deceased patient. So you see this brain is a bit different. Maybe it's missing some white spots here. So I'm reconstructing a healthy guess of the deceased patient. And then I can just do the subtraction and it will highlight my anomalies. Okay, so that's the second way, a second popular way of doing anomaly detection. So what's the difference with this one? With this one, you only get one value. Here, I'm just saying how far is my red dot from the greens. It's just one value, how abnormal is my new case. In that one, I'm getting a full map, an image, a picture of where are my anomalies. So it's a much more complete picture of my anomalies. Okay? Okay, so for the next part of the session, I will talk about what is a representation of the data, because you've seen in my diagrams, I'm using a lot this, encoding, decoding, basically to go from an image, from high dimensional data to a simpler representation of my data. So the key step is finding a data representation. And that's the second part of the talk. We'll talk about how to understand the data. The key word is low-dimensional embedding. We'll quickly talk about some statistical meaning to it, and the reconstruction, basically, which is the decoding. So representation learning, it's basically better understand the data, the space from which your data comes from. So you look for a space of lower dimensionality, and you look for this without any labels. So my inputs, they could be this in medical imaging. They could be a single image. But by single image, why is it high dimensional? Because there's potential information at each pixel. It could be also multiple measurements. You could see as a long vector of data, or multiple images, multiple high dimensional images. So this is somehow complex data, and I would like to simplify my data to come to a space where each dot is a subject. And if you see here, this is one subject that I'm able to represent with much less dimensions, d dimensions. So there's 3 steps for this. The first one is encoding the information, which I'm talking about embedding. The second thing is which properties I would like for this space of lower dimension, which we call the latent space, and then being able to reconstruct my data in our field for medical imaging, because I would like, if I provide the cloud of points to the clinicians, they'll say, I don't understand this, it is not talkative to me. While I understand as clinician the medical images. So a bit of notations on this. I will represent my input data as X. My data may lie into a specific space, which may be nonlinear. We'll come back to this. I would like to estimate a space of lower dimensionality, which I call the latent space. And the key thing is these spaces have much different dimensionality. The input space, if I have an image of 100 by 100 pixels, then it's 10 power 4 dimensions. And my latent space, maybe it's going to be 16 dimensions, 32 dimensions, which is not a lot. A key concept to start with is, should I do linear or nonlinear analysis? If you're in physics, I think this may be talkative to you, but not necessarily for images. So suppose I have 2 images. Here, these are the pictures of 2 PhD students. I would like to have the average face of these 2 subjects. So basically, these are my data. Let's take a point which is in between. Let's do a linear average of the 2. So that's the linear average. The linear average is at each pixel, I combine the information. And this is no longer a face. It's a mixture of the 2, and that's not the result I would like. So the key thing is to say my images, and that's the same for my medical images, they necessarily not belong to a linear space. So you see here, that's the space of my data, the donut. And if I take the linear average, I'm out of the donut. So basically, I'm getting an image, but that image does not belong to the space of face images. And what I would like to do is basically find an image that belongs to the donut, and that's it. So that's the nonlinear average. So back to linear. Obviously, there are the simpler techniques, and I think you may know, who knows this one, who heard that one, principal component analysis. Okay, few people. It's very useful for any type of domain. And obviously, it's very simple. It's linear. It's very useful to start with. Okay, this is the nonlinear thing is when you already know you work with complex data, you work with images, you work with objects for which you have a specific statistical properties. But start with linear. So principal component analysis, the basic idea is you work with something which is the covariance matrix. So the covariance matrix, it's a matrix that has as many dimensions as the dimensionality of your data. If you work with scalars, the variance in your data, variance is a single value. If you work with 2-dimensional data, somebody talked about strain just before, strain in 2D, it's 2 by 2 matrix, it's 2 dimensions. The same will be for the covariance matrix. So this is a matrix which is symmetric with real values, so you can diagonalize it. It means that you can represent your covariance like this. Who knows about diagonalization? Who heard about diagonalization? Not so much, yeah, a bit. So basically, when you have a squared matrix with real values, you can find another representation where this is a diagonal matrix, D, and P is just, you can see it as a change of basis. And in that new space, you have two objects of interest, the eigenvalues that represent the variability in your data, and the eigenvectors, which are basically the change in dimensions. So an illustration of this, it's this space. I've taken a space which is like this. This is my data. So basically, it's a linear, it's a plane, but in a 3D space. So 3-dimension, it's my high dimension in that example. Okay, but actually, I just have 2D data. That's my sheet of paper that I would like to estimate. So here, the picture is my 2D thing into the 3D space. If I apply PCA to it, I get this. So actually, I'm able to estimate the main variations in my data. Okay, so first variation, the second variation, and whatever my orientation is here, I'm able to recover this. Okay. What for? Basically, I'm guessing what's the organization in my data, and then I can move across my space to see what is encoded in my data. So the center, it's the average in my population. Here we did this with cardiac shapes, so this is a heart. That's the average heart. And then I can just navigate across the first dimension. I can navigate across the second dimension and try to reconstruct, to decode cases to see what they encode. So that's one. These sample here are decoded, and I say this is what is encoded across the first dimension. If I navigate here, I'm just reconstructing samples, and I see that my first dimension is encoding these properties of my heart. It gets bigger, etc. I could do the same for the second dimension and just generate samples along that dimension, and you see that my second dimension is informative of all the properties of my data. It encodes, in that case, different aspects of my cardiac shape. Okay. So when we go nonlinear, obviously, it's a bit more complex. Nonlinear means my data is not organized like this, but probably like this. So it's still 2D data, but organized according to my donut structure or to another structure. And still, I would like to recover my 2D embedding. So the key thing is the structure of the data space that I've shown you before. A key word that you may keep in mind is what is called manifold. A manifold is basically, or a simple way to see the manifold, it's the Earth. It's something which is nonlinear, but when you look closely, when I look at the Earth, locally it's plane. It's a plane. So that's a good example of a manifold in that much dimensions. So in some cases for strain tensors, you know that they belong to a given manifold. And that, in that case, the manifold is known. For machine learning, we need to learn it from the data. There's many ways to do it. It belongs to the work of representation learning, manifold learning. The first thing you assume is your data belongs to this specific space. You have few samples available across that space. And then you try to link them. You try to say, okay, that this subject or this sample looks close to these ones, etc. So you construct a graph across your data. And once you have a graph, means you may have a matrix, a squared matrix. so you can diagonalize it. So you, again, there's this step of diagonalization and basically to estimate that space. So that's the common recipe to do this representation learning. A quick illustration of one very popular example. It's from 2000, but still it works well. People looked for the variability into brain images. And the same example as before, if I average 2 brain images, it's no longer be a brain. While if I use a nonlinear embedding algorithm, for example, this one, which is called isomap, I can get a representation of my population, and then I can navigate across the first, the second dimension. And here you see all these images. This is the average. This is the first mode of variation. The other one, you see tiny structures that vary across the images. Okay, the ventricles inside, some kind of patterns outside, and they all look like brains. Fortunately, because these people use nonlinear methods to do this. Another way, and I will maybe skip the equations, but just you can come back to it later. Another popular algorithm, it's called Laplacian eigenmaps. I'll skip again this. The key thing is it involves the Laplacian of the graph. And basically, if you're from the physics, Laplacian, it means second derivative. Okay, and when you look for the Laplacian, it may sound to you like vibrations into a material, vibrations of a plate or something like that. So here I used this algorithm again on my example. So that's my nonlinear space into, that's the 2D nonlinear organization of the data within my 3D space. And with this algorithm, I'm able to unfold the data and get this estimation. So again, that's not so nice, but still I get a nice organization from blue to red across the first dimension and from dark to bright along the second dimension. So that's a nice algorithm, a nice way to do it. Another way to look at it, and if you work with materials, you may be familiar with this. If this is my nonlinear space, I can look at the first variations in my data as this. So you could see it represents the length of my material. The second one is the height of my material, and the other ones are harmonics. Another way to see it is like this: the first mode of variation, the second mode of variation, it's a harmonic of this. And when you have more complex data, then you get much more harmonics. You could see it as the modes of vibration of your material. This is what people got with interferometry without any machine learning. They had a metal plate and doing interferometry, and that they got these images of the modes of vibration. And that's what people got with the representation learning I've shown you on brain surfaces. You see some similarities. You see the modes of vibration of the shape of your brain. Okay. So in brief, there's many algorithms. I don't want you to retain this. The key thing is how to represent complex data, choose between linear and nonlinear, and you fix an objective. The distance between my data is something, the geodesics, anything. You can see popular algorithms, PCA, I've shown you before. There's a unified framework. I'll skip a bit this, but the key thing I would like you to retain is all these methods, they work with a graph that links the data. And basically, they diagonalize a matrix of interest. So for principal component analysis, the matrix is the covariance matrix. It means you work with the principal modes of variation. Laplacian eigenmaps, I've just shown you before. I told you there's the Laplacian involved. So you diagonalize the Laplacian. Isomap, it works with the geodesic distance. So you diagonalize a matrix of geodesic distances, etc. Okay, so back to our pipeline. We were trying to represent complex data into a very much simpler space. So basically, we were trying to do this. Try to find a function that maps x to y, keeping in mind that we have high dimensionality and low dimensionality. And I've told you that if I show to a clinician a space like this, they'll say, I don't understand, I don't care. What I would like to see is images. So the key thing is being able to reconstruct, so to go from my low-dimensional space, any sample, the average, the modes of variation, and being able to reconstruct images from this. So basically look for a function that maps from y to x. So with PCA, it's linear. It's very easy. I've told you that when you diagonalize, D, it's the matrix that says my modes of variation. And P, I told you that these are the eigenvectors, which can be seen as a change of basis. So basically, I can go from X to Y by just a matrix multiplication. You could think of a rotation if you'd like to simplify. A rotation is a matrix in 2D. It's a cosine, sine, minus sinus, and cosine. So you just apply the rotation matrix and you move your data from X to Y. Here, it's a bit more complex, but still you could see it as a change of basis. And basically, if you take the transpose of this matrix, then you can do the opposite operation. So with PCA, it's easy. With nonlinear algorithms, that's not easy. You need to do interpolation, etc. So it might be very complex. Unfortunately, and that will be the last part of my talk, there are much newer methods that are called auto-encoders that do both the encoding and the reconstruction. And I like them because they allow many extensions. So I think it's worth taking some time to discuss them. So that's the idea of the auto-encoders. They correspond to the schema I've shown you before. So you have an image. Here it's the image of a digit. You would like to encode it to a very simplified set of variables. If here I encode my image, I don't know how much pixels, but let's say 64 by 64. And I'm encoding it into 1, 2, 3, 4, 10 values. So my high dimensionality is 64 times 64, and my low dimensionality is 10. And only with these 10 latent codes, I'm able to decode and reconstruct an image, which is not, you see this 4 is a bit blurred, but still it looks like the original 4, okay? So that's the basic idea of the autoencoders. You basically learn both to encode and decode your data. So you need a loss function. And basically, the loss function will be the reconstruction error. You will say, I'll compare my output with respect to my input, and I'll try to maximize this. So I'll try to optimize the parameters of my encoder, to optimize the parameters of my decoder, such as I have the best reconstruction. That's nice, but that's not maybe the best for handling populations of data. So with autoencoders, basically you consider your data with a few parameters. Let's say this is an image of a tiny boy and the Joconde. If you say my latent code associated to them, this little boy is sad. So in the scale of sadness, it's here. The Joconde, I don't know, but maybe it's neutral. It's in the middle. And then people invented the variational autoencoders, which are much better for representing the data. So basically, the little boy, I know he's sad, with a bit of uncertainty, but in the scale of sadness, it's here. The Joconde, I don't know, either she's smiling or not. So she's centered around a neutral smile, but with a large variability. So the key idea is, instead of encoding your data, encoding your data with a single code, you encode it in terms of a probability, namely a value and a variation around it. So that's it. You encode the data, you decode the data, and all these attributes, here they are called smile, hair, gender, etc., but actually they are just latent dimensions, 1, 2, 3, 4 until D. You encode them in terms of probabilities. So you have two pieces into these methods. You have an encoder, a decoder. So they belong to the class of autoencoders. So you have a loss for which you would like to minimize the reconstruction error as before. You compare the output with respect to the input. And the second thing, you add a term such that there is some consistency into the latent space. Basically, the distribution should match a Gaussian. So basically, there's a loss. Here, KL stands for the Kullback-Leibler. Basically, you say the distribution of my data here should look like a Gaussian as much as possible. So I'll come back to this afterwards. Oh, no. Let's say encoder and decoder might be mysterious to you, but basically the encoder, it's a neural network, okay, that relates the input variables, so many variables, to a few variables, and I told you they are encoded into probability terms. So basically you encode the mean and the variance. And the same, the decoder is a neural network that maps you to the output image. Okay? So this is simple neural network. For images, we can do it with convolutional neural networks, the same thing. So if I only use the reconstruction error for my problem, let's say my data, they are the digits like this. So I have 10 classes. This is my latent space. So this is a simplified view of all the possible digits written by anybody. So you see that if I only care about the reconstruction, they are very clustered. Okay, I clearly see this one, which is digit number 1. This one is digit number 6. Obviously, some look like the others, so they are mixed. But you clearly see some clusters. If I only use the other term, if I skip the reconstruction, I only focus on the fit to a Gaussian distribution, obviously I get this potato, like everything is mixed as a Gaussian, and there's no separation between the two classes. And if I do the good way with a good weight between the two terms, you could see there's some kind of consistency, some kind of Gaussian organization, and I also distinguish the clusters in my data. And that's very nice because here I have a consistent space into which I can navigate and look at what does the first dimension encode, what does the second dimension encode as before. Okay? The bad point about the variational autoencoders, VAE, is that the reconstruction is nice, but not so nice. So many people are working with this with images of faces, and you see that these are some faces that have been sampled from the space, the space of images of celebrities, and they are blurry. Okay, you can still guess some people, but they are blurry, the background is blurry, etc. And what about having a better reconstruction? Now people, when they do image generation, they use this, which is diffusion models or other techniques. So that's the same idea. You start with a low dimensional space, but then you get much better reconstruction with some techniques I won't detail. But keep in mind that if your objective is to get much better reconstruction, then this is a family of methods to go for. Okay? So as a conclusion, and then I have some slides about this, and then we have some time for questions. Self-supervised learning, or unsupervised learning, it's very important to keep in mind. Many times people think of supervised because I want to do classification, regression. But supervised learning, it's relevant when you don't have labels, when you don't trust in the labels, or when you just want to look at your data without being influenced by your labels. And generally, I like to start with this. I have brain images, cardiac shapes. Let's just go to what's the variability in my data. The same as when I look at you, I'm thinking, I'm not classifying who is a girl, who is a boy, who is old, who is young. I'm just looking at the variability of your faces, etc. Okay? Then obviously it depends on what's the question you'd like to address. And I think clustering or anomaly outlier detection, they are very popular questions to think. And so unsupervised learning is very relevant for this. And if you have something to keep in mind about this is it's key to learn a representation. Okay, so to encode your data to a space which is relevant. And for this, you need to think of linear or nonlinear. Start with linear. And it depends on your objectives. If you'd like to think of meaningful distances between your data, geodesic distance, statistical distances between your data, then manifold learning may be nice. But you've seen that the reconstruction, not so nice. You need to do interpolation, it's not optimal. Otherwise, the auto-encoders, they are very nice because they, by definition, they consist of encoding and decoding. Namely, they do the reconstruction, but they have limited statistical meaning, even the variational auto-encoders have shown. The other questions that are related to unsupervised learning for any field, we don't have labels, so they are very hard to validate. Classification, it's easy. I have a training set and a testing set. It's very easy to derive some metrics to say I'm right or not. For unsupervised learning, that's not easy, and it may depend on your objectives. If you do risk analysis, you may still find ways to validate this. And I would like to insist on unsupervised learning because I think it's underused. It's very promising for medical problems, just to look at your data, to understand your data, to communicate with the clinicians. Okay, so it's a very good tool for this. And what's next behind this? I've not talked about multimodality, but obviously that's a component of what we do. We look for the analysis of brain images with MRI, with PET or anything, and we may mix this with the patient information, the age, the sex, the height, etc. So that's by definition multimodal data. I've not talked about the uncertainties, but there's uncertainty on the data, there's uncertainty in my model. So basically, if in my representation, I'm not sure that the patient is exactly like this. So that's a field of interest at the moment also to model. So that's it. I hope I've given you a broad way of unsupervised learning, self-supervised learning, and I'm happy to answer some questions about that.